There's a hair on the end, is that right?
There is a hair.
It's meant to.
Eyelash. It's meant to be.
Oh, it's meant to be.
Yeah.
Prince William recently stepped inside the Francis Crick Institute, a world-renowned biomedical research powerhouse in the heart of London. Ahead of the centre's 10th anniversary, the Prince got a VIP look at the future of medicine. He spent the day touring high-tech labs, including a rare look inside one of the world's most advanced imaging facilities. From chatting with students to diving deep with neuroscientists on how to restore human speech, William was clearly moved, calling the life-changing work incredible. Stick around to see exactly what happened when the future king met the minds changing the world.
We really just trying to understand that is you look at the structure, right? You know, the computers, we look at the wiring, the components, as well as its function. And on the structural side, we actually got sitting over the last decade, it's like millions, it's the millions of things. In order to go through a single memory you can tens of thousands of those. It makes it very difficult to large you see how all these cells are wired up. You see all these fibers that connect between individual neurons. We hope that these approaches will allow us to understand how brains are wired up, how they're differently wired up in different disease conditions as well, and maybe also learn about natural intelligence and how it actually comes about from wiring. But that's one side. The other is really the function of the brain, the electrical activity of all those neurons. And we and other groups develop kinds of technology to put in wires into the brain, some to just do our research in the lab, but also, one startup coming out of my lab is now. And my lab itself, I have 2 neurosurgeons as well. It's one of the benefits of the Crick, really working very closely with the clinic. And those surgeons are now putting tiny wires into the drives.
Presumably if we can understand more of the wiring, we can also, obviously the disease part of speech yourself, but in terms of our emotional regulation and that stuff, because that helps understand mental health and what stuff too.
I'm a very discovery scientist. Yeah. You have to really understand.
And what is often like, and more often than not, hello! Very nice to see you. And you. So, welcome to Electron Microscopy Science Technology Platform. So, I'm Deputy Lead here. And what we do is we work on routine projects. We have quite complex projects, and projects last between 6 months up to several years. And they're all pretty much bespoke. We have lots of different types of technology, and they work very closely with the group leaders. So the sort of samples that we use, we look at cells, so the basic unit of life. So it could be individual cells, or cells obviously sort of forming tissue, which is you get, in your biology textbooks, you get pictures of a cell. That was obviously happening.
Just my biology A-level.
Nucleus, mitochondria.
Yes. Yep.
Good. That's the sort of thing you get. And what we're going to do in a second, we'll go to do in a second, but in order to get the samples into the microscopes, the inside of the microscope is very inhospitable. It's high vacuum, we bombard it with high energy electrons, so we have to do an awful lot of sample preparation because we don't want our nice little soft biological material going into a vacuum. It doesn't tend to happen. So we have a whole huge process with chemicals, etc., etc. But for samples that go into the TEM, which we're going to look at in a second, we have to then cut very, very fine sections, which is Dumi. She is going to tell you. So this is Dumi Nirvana.
Hello.
She is
that was funded by the CCI. So that was then to build a pipeline where we democratize access to. So the point is that they can be used in low-cost settings or in remote facilities where people cannot get to put expensive machinery or change the environment to hold them. Used there for local research. So because I don't have much time to get more into details on that, the fun part begins. So I'm going to need your help.
Sure, yeah.
So
Make sure your fingers don't get cut off.
What's actually in there? What are we looking at?
So this is my block, and we're ready to section it. It can be anything, it can be brain tissue. Upstairs you already heard about the connectomics project. So all of that can be cut and then basically put together again after you image to create a volume. So you're kind of like working backwards. But the point is that you can then put all of these together to create a volume. Most important is that you pick them up in a serial format, so you need to be able to look under the microscope and know which one came first, so then you can pick them from 1 to 1,000 or whatever ridiculous number, 100,000, because depending on how much of the tissue you have. So these ones are all over the show. So if we press here, so I've set the start and the stop for the microtome basically to kind of like slow down as it hits the knife, because you don't want to damage that and you want to cut it nicely. So sometimes it takes a while to basically produce a section, but we already have ones cut for you. So what I will need you to do is take a sip, and then you can adjust then the eyepiece so that you can be able to see and keeps cutting. But if you stop it, it takes a while to again start like cutting. So it's either if we can wait for that advance, if we run out of patience, we can move along.
This is 40 nanometers.
So these you can cut down to 30 nanometers.
30.
Yes. So if you think of a cell, which is like probably can go to 10 microns, and then you have to cut it at 30 minutes. So you're going to end up having like 330 sections that you need to pick them up. If you're cutting the tissue, then it's a whole lot. Some people do a whole worm, which is going to be like thousands of sections that you can have. I think there you go now. It's about to cut.
Are they done?
Yes, until it is done. So now that we have quite a few, this is your eyelash.
Okay.
Now, what I want you to do is moving the sections.
There's a hair on the end, is that right?
There is a hair.
It's meant to.
Eyelash.
Okay.
It's meant to be
Oh, it's meant to be there. When you said eyelash, it literally is there.
It really is an eyelash.
Okay.
Yes. We are very obsessed with eyelashes. Everyone with long eyelashes, we're like, can we have some? Yeah. So we're going to stop now, and then you can move them. The only thing that is important is that you don't poke the sections because they can tear up.
Okay.
And also sometimes they curl around the eyelash. Okay. So what you want to do is either you maneuver them at the corner, or you use the water to roll it back so that
You can tell me this is like a really serious research project you're doing.
We have a replicate. Don't worry.
I'm not responsible for messing this one up.
Yeah. So here we have an ITO.
Okay, so I'm brushing them back.
You're brushing them back in a sequence, because I want them
all together?
all to the back.
Oh my. All together, they can't break out.
You can break them up as long as you can put them together again in a sequence.
Well, I'm moving on in a sec. Okay, let me see.
She's a hard taskmaster, isn't she?
It looks so weird in the. Okay. Is it best to have it facing upwards or downwards?
Gosh, whichever way.
The ones that do.
Okay, so these are the students from William Ellis School. My colleague Ellie here is going to introduce you to the ones that are going to be chatting with you today.
Super.
Do you want to take a seat? We've got you some gloves, we've got everything ready.
Excellent, ready to go.
And I can introduce you to, we've got...
Nice to meet you guys. What are you learning today? What's going on?
PCR.
PCR. Is that something to do with swabs and COVID? I remember PCR from COVID days.
Yeah.
Is that?
Yeah, basically that.
Is it the neutral sample? Is what it's called?
Neutralising?
Is it neutralising? Is that one?
It's not that one.
Okay.
But
What's the PCR then?
Polymerase chain reaction.
Okay. Can you just tell me what it?
You've just modelled it over there.
Yeah.
So what are you doing in the model?
So you're breaking up DNA chain and then primers attach to the DNA and like it's all so that we can use this in the production of insulin through E. coli. And originally it was produced from pigs and how we use the bacteria due to ethical reasons and
Amazing. And so what do you have to do? Do you have to actually draw it out of a substance, or is it to go into the little pipettes?
Yeah.
I think this is where you get your chance.
This is where I have yeah. As well, okay. You should.
So you take a microfilm.
Yep. Goggles, yes, glasses. I'll get a glove on first, then put the glasses on.
And sorry, just like to remind you. I'm sorry.
Glasses on.
So you take a microfilm.
Yep.
There's 2 stops on it when you press down to the position.
Yep.
You press down to the second stop, and that dispenses it.
Okay.
And then the white tab here, you do that to get rid of the tip. You press that to get rid of the tip.
Gotcha. That's it. There. It's like this in there.
Yeah.
Gotcha. Press the.
Don't use anything.
Just like that. Okay.
It's also best to hold it.
Or you go and hold it to the first.
Push the. Here we go. And then release.
Yes.
Okay.
And then take it out.
Aha, we've got something good.
And then place it on
here. Pressing back in again.
And press it all the way through.
All the way through. Yeah.
There we go.
And then use that white to get some.
I get that.
Yeah.
Hey. Okay. Thank you.
Perfect.
So what is that apple under? That's dye, is it?
This is just
Food dye.
Okay.
That was just a practice earlier with some food dye.
Oh, okay. Yeah, I've not trusted anything too special today. It's good news. Look at the electron microscope downstairs. That's like quite a big bit of kit, quite a serious bit of kit. Don't get me touching that. Getting expensive. Are you enjoying learning though, guys? Are you all into science and kind of research?
Yeah.
Okay. What do you all study?
We're all A-level biology students at the moment.
Where are you all the same? Where are you at?
We're all at this one.
Good. So actually coming here and doing the practical work is quite exciting, quite interesting.
Yeah.
Do you get to do much practical work where you are?
Yeah.
You do. But here's different because
Well, we get
more stuff to do, more fancy equipment. You get Charlotte helping out.
Yes.
Good. And Charlotte, what sort of things do you try and expose them all to that they may not get a chance to do elsewhere?
I guess these kind of pipettes here are quite interesting. There's different amounts.
It's a vortex mixer.
Yes.